All right. It's my pleasure to introduce Dr. Bob Klein. Bob is a graduate of the University of Wisconsin-Madison. Received his master's, PhD from the University of Illinois. After postdoctoral appointment next door in BioBio, where he learned molecular biology from various entities there. Bob joined the USDA-ARS in 1989 in Lexington, Kentucky. And then in 1997, he felt he had to get back to Texas, and so he joined the ARS team here. His research focuses on developing the genomic resources to assist sorghum breeders as they labor to improve sorghum grain and forage yields. And Bob is colorblind, and he's also a sailing enthusiast. So there's some extra information you didn't think you were going to get.
I did not write that down, John.
So I'm okay with this mic for the people online just sitting here? Okay. So first of all, thanks for inviting me. Maybe I should give a little introduction what I mean by my endless project. When I arrived in 1997, my predecessor, Keith Schertz, was a heck of a geneticist, and I admired what he did. And he had a great interest in cytoplasmic male sterility and fertility restoration. And to continue his legacy, I thought I should continue on his work. Unfortunately, I thought I would continue it for 5 to 10 years because I thought the answers would come easy. And some 24 years later, I'm still dabbling in it. It's not my major focus, but I want to get to a point where the project is complete. But never did I anticipate I'd still be working off and on this project after all these years. So, okay, I can't go back. Help me, Scott.
You have to click on the screen first. And then you can find
How
For
do
the
I-
mouse. For the mouse.
Trying to,
click on it once. And you should be good.
There you go.
okay, to go back is this one. I'm going forward. Okay, that's where I thought I was speaking from. So the question has been through this project is, do I continue or terminate? And I think most of you know my plans of leaving in a reasonably short amount of time. And one of the reasons is things like this, hiking in the Colorado Rockies in the middle of winter with my son. And as you can see, you even have a case where a hungry bird landed on a branch near us and decided it wanted some of his granola bar. So the things I truly enjoy doing. So that goes to the continue or terminate statement. So a little bit about cytoplasmic male sterility. First of all, in sorghum, it's critical for hybrid seed production. However, I am not going to sit here now and tell you what we determine with cytoplasmic male sterility or the nature of the RF genes is going to assist breeders. That is something we were saying back in 1998 through the early 2000s. MapQTL is going to help breeders out immensely, and realize at this point that's not my reason for continuing this project. But as Bill Rooney once said, whether what we find is critical, whether it be in black sorghum or CMS systems, whether it's critical to the breeder isn't necessarily the point. It should be our main focus, but we're also scientists and we have basic curiosities. So for me, that's really why I have continued this project, is because it is a fascinating story of genome evolution. And what I mean is the mitochondrial genes have many defects, and the nuclear genome has rapidly, in an evolutionary sense, rapidly evolving genes called PPR genes that actually get transported into the mitochondria, the protein does, and it actually corrects the mistakes. So if you look at the mitochondrial genome, you rarely see SNPs. You mostly see massive genome reorganization. And it's these fertility restoration genes and other genes within the nucleus that go in and correct those defects. Now, where did cytoplasmic, what leads to cytoplasmic male sterility? After all the years of working on this, we now know it's basically just an incompatibility of the nuclear and mitochondrial genomes. Okay, and I'll go through a little bit more to introduce that. So as it says, we realized that cytoplasmic male sterility, at first they thought it might be the chloroplast, but it is the mitochondria. And the nuclear fertility, nuclear encoded fertility genes are what actually rescue the function of the mitochondria. And they're mostly out of a class of genes, which I'll get to in a minute, called PPR genes. But again, it's because they co-evolved that you're not going to see the sterility most times within a given group of sorghums. You have to look outside the group to be able to make crosses that reveal the cytoplasmic male sterility. So what's interesting about sorghum, if you read the literature, they'll talk about rice and other crops that you have to make a wide cross. And through this wide cross, or very distant relatives, you discover cytoplasmic male sterility. Ironically, for sorghum, it's very different story. These sorghum lines that were found that led to the development of the A1 cytoplasm being discovered were actually land races or cultivars that already have been grown in the United States from the late 1800s. But if you'll bear with me for just a second, because I want to show you how this might have taken place. And if you'll see this hatched area, that's about where the center of origin of sorghum resides. One of the very first things you note is that then sorghum was moved to Ethiopia, which is presently Ethiopia, and that's where the race Duras developed in approximately 2,700 years before the present. Why is this important? Well, Milo sorghum is a Dura. Now, if you grew up on a farm, you always call it just Milo. Well, for a geneticist or a breeder, Milo refers to a specific group of sorghums that are a subset of the Duras. So they were developed in Ethiopia, in that portion of Africa. Also, at the same time, you had sorghum being moved to the west coast of Africa, and then the Bantu people carried it south. And as they carried it, they developed sorghum, is what they took along with them. And through their efforts to move to South Africa, you have the development of the race Kaffir. So here's the important ones, the Milos and the Kaffirs. Now, how did they get to the United States? Most likely, a lot of it came via slave ships, especially, I believe, the Milos, because if you notice over here, I said the history reports say that the Milo came to the United States via Colombia in 1879. And you go, wait a second, I thought it developed over in Ethiopia. Well, I think it got left over and it made it on a ship, if I understand right, to South Carolina, then eventually over to Texas. By contrast, what I understand about race Kaffir, it was introduced somewhere in the late 1800s, mid to late 1800s. What's interesting about this, if you read the history, the seed of black hull Kaffir or Kaffir sorghum was actually on display at our country's centennial celebration in Philadelphia. So it was attracting attention. But where does cytoplasmic male sterility come from? And what it was is the people actually at the Texas Egg Experiment Station, they recognized that other species had cytoplasmic male sterility, and they realized the value of this in making hybrids. So Stevens and others started making reciprocal crosses. And ironically, by crossing a line that you could find in a farmer's field, dwarf yellow Milo, and of course, Texas black hull Kaffir, by making reciprocal crosses, they realized that they had cytoplasmic male sterility existing within the Milo cytoplasm. And for the breeders out here, that's your A1 cytoplasm. And the history reports, Stevens sent a letter and said, we have it. So in about 1950, they realized they had it. Okay, and this is, as you can tell, these aren't wide crosses, right? It's two domesticated lines. But when you put the Milo cytoplasm with the Texas black hull Kaffir, they evolved in completely different regions of Africa. You end up with incompatibility of the nuclear genome of Kaffir and the mitochondrial genome from Milo. You get cytoplasmic male sterility. Okay. So just to summarize the historical perspective again, A1 cytoplasmic male sterility was discovered in Milo, and they just determined this by doing a reciprocal crosses. The reciprocal crosses told them they didn't have genetic male sterility, they had cytoplasmic male sterility. And again, as I've said, that's due to the incompatibility of these two genomes. But then they said, where do we find our restorer genes? Well, of course, you go back to the Milos because Milos aren't sterile when you cross a Milo to a Milo. So they determined that their RF genes resided in the Milo nuclear genome. So basically, the system had been established, and maybe I think about, Bill would probably correct me, sometime in the mid-60s. Hybrid sorghum was very prominently grown in the United States, and it all started at the Texas Egg Experiment Station. Okay. So I'm going to give just a brief background, on what causes this cytoplasmic male sterility. And the easiest thing to recognize is this. There's dynamic genome reorganization. And for those that work on a nuclear genome, we would call them structural variants, okay? Not SNPs, but massive reorganization. And how the heck does this take place? And if you look over at this figure, where you see little tick marks, there are repeat elements. And what happens is you get crossover events. This would be like a mitochondrial chromosome. You get a crossover event there. And what you end up with is two subgenomes. They're all still made up at the same pieces, but they're not in the same order as over here. So why is that critical? Well, at this given point where I have the cursor, you may have had one gene, and that gene went, if I'm correct, from the yellow to the black area, with one end of the gene in the black and the other end of the gene in the yellow. When that gets reorganized, as in over here, all you have left is part of the protein in the yellow part, but there was probably a gene in the blue as well. So now you've got a chimeric gene, and what you can call it a centaur or whatever you want. It's got the head of a person and the back end of something else. Okay, so these are literally chimeric genes. What's the important? Well, these novel open reading frames, where you've got one gene fused to another gene, are believed to be toxic to the mitochondria. But here's the catch to that. You might go, oh, I remember taking Scott's class. Mitochondria are important in the seed germination. They're important in the roots. They have important functions. Well, if it was in all the cell types, it would be a lethal mutation. So I'm sorry, photosynthesis is not taking place in roots if they want to have energy. They're using mitochondria. So there's something very unique. You can't just generalize and say these chimeric open reading frames are toxic and by God, it kills mitochondria. If it did, the plant would never grow. It'd simply be it would simply be a lethal mutation. But because of all these reorganization events, you end up with chimeric genes. And starting about 10 years ago, people are like, could these chimeric genes be the thing that are causing cytoplasmic male sterility? And in essence, that's exactly what's going on. So when I first started this project, maybe one or two of these were known. Daryl Pring had done that. They knew, the Lonins knew about the T cytoplasm, but there was very little known. But through this time, we've learned, and we had our candidates years ago, but I wanted more proof before we published. But here are some of the features. What you find for these toxic mitochondrial genes, as I just said, they're a portion of one gene with some other sequence on the other end. Here's something that Rai Young, who's in bio, would say, that one of the reasons why they're toxic is when you fuse these two different proteins together, in the junction point, you often get a transmembrane spanning domain. And you go, yeah, who cares? Well, what happens is most of these things, like the ATPase subunit 1, does not cross a membrane. But the chimeric gene from it does get stuck in the membrane. What Rai Young used to say is called the constipation hypothesis. That people, when we first started in molecular biology, we tried to overexpress everything in the world in E. coli. And many times it was lethal to E. coli. And the reason why that was because it would have a transmembrane domain. And he says it's getting stuck in the E. coli membrane and it's killing the E. coli. And so a lot of people feel this transmembrane portion is very important to be toxic to the mitochondria. You're looking for a nice clean membrane, and here you have this foreign portion of a protein stuck in the middle of the membrane. Again, somewhat speculative, but some people also in certain systems have proven that. And just to finish on the bottom, as I said, my feeling and others that it's got to be a specific cell type or a specific stage of development, because it was happening all over. You wouldn't have very good germination without functional mitochondria. And that's actually one of the reasons why it becomes hard to work on. So starting about 10 years ago, before all the great technology now exists, there was no PacBio, there was no Minion long-read sequencing. What do you do? Well, starting about 10, maybe 12 years ago, working with a company, we went back to very old technology. If you wanted to work with something big, you put it in phosmids or back libraries. So we put it in a phosmid. And basically what this does is it gives us long read order, because the smallest insert size was 40 kb. So we know if we sequence from one end to another that we know the order, which is important when you have massive genome reorganization. So we did all that sequencing. And then the new technology came around and we realized that, well, let's confirm our assembly using the Minion. So once again, I went into mitochondria, isolated DNA, and it did prove that what we had was accurate. We weren't missing any pieces, but the new technology, you go into the literature right now and it seems like people are sequencing a mitochondrial genome a week because all they do is they isolate mitochondria, do PacBio sequencing, you get nice long reads and you go, there's the sequence, and they quickly annotate it and publish it. Maybe I should have just waited to do the project until 2020. But here, just graphically representing what is going on with these mitochondrial genomes. And the only reason why this is critical is when you look at the junction point of some of these fragments, like where I have a cursor, most likely you're going to have a chimeric gene because one end of the gene was on that fragment and another end was on this. But the easiest way to look at why this is complicated, you see this piece here in subgenome 3, which is identical to this, down to a nucleotide. But it's the reorganization and the new fusions that lead to these chimeric open reading frames. When we were putting this together, the best way to describe this is these are unique. Picture this a jigsaw puzzle. Through here, these are all unique pieces, that solid black line. But all these other ones are perfect repeats, but they're all assembled in a different way. So you have in your jigsaw puzzle, you have 2 or 3 yellow fragments, which are exactly the same shape, what they contain, but they don't go at the same place within your jigsaw puzzle. So it sounds easy, but it gets rather complicated because you could be going down this yellow thing and then all of a sudden hit the sequence for this long region here. Again, these are all gene-rich regions, but down here you end with the yellow fragment and you hit something completely different. So it took a while for us to assemble it, pardon me, but we got it. Suffice it to say that the whole reason for doing this is just to try to find what gene within the mitochondria could be causing this cytoplasmic male sterility. The only reason why I put this up here is I always thought it was kind of pretty. Everybody does them, it's the alignment of the genomes. The only thing I expect anybody to come away with, when you look at the normal mitochondrial genome, what anybody in the Sorghum group, it's BTX623, okay? This is the genome. It's one circular molecule, period. Okay, then you come over the A cytoplasm and you have 5 subgenomes. The DNA in the normal cytoplasm is 99% of it is represented within these 5 subgenomes, but the order is completely different. And what this is a ribbon diagram. As you can see, this piece goes there, you have this going over here. It's so much reorganization. If this was not much reorganization, all you'd have is one big ribbon going out and saying you're identical besides a couple of SNPs. But this is what has made sequencing the A1 genome so difficult, is because of that massive amount of reorganization. We were basically doing, you always see this term now, de novo assembly. Well, we did de novo assembly because aligning it this versus the normal genome, all it did was confuse the hell out of us. Okay, there's just that much reorganization. But who cares? And the reason why you care, you see I've labeled a few genes here. These are all chimeric open reading frames. Okay, again, the centaur protein, they don't know what the heck they are. And some people believe they're toxic. But as you look of ORF open reading frame 624, it occurs right at the fusion of these 2 fragments. Okay, and so you can go here, again, here's a chimeric gene. Again, it's right at the junction of these 2 pieces. So again, it's like jigsaw puzzle where you're moving parts around, and where they align in the jigsaw puzzle gives you a completely different picture. And in this case, it gives you a completely different gene. So after all that hard work, which if we had PacBio would have taken us about a month, we have a complete annotation of a genome. We have candidate genes for cytoplasmic male sterility. Again, I'm not going to tell you this is going to make plant breeders' job any easy. That's not the case. It's just, I didn't want to give up on this because a lot of people had worked on it. So I kept dabbling. But this is a graphic representation. We're not going to go all the way through this. This is already prepared for a manuscript, and I just left it all on there. But I'll show you the chimeric structure. This right here, ORF624, we believe is the gene causing cytoplasmic male sterility. Why? Okay, first of all, it starts with a very critical ATP subunit. It matches it perfectly up to amino acid 484, and then it's fused to something because that's right at that junction point. And the closest homology we get is something called ORF 182, and it's from rice. We don't even know where this hunk is. It's in the genome. in the genome, excuse me, but we don't know how it got there. I also told you, this is right here, these areas here are going to tell you whether you have a transmembrane domain. ATP1, no transmembrane domain. You go over to ORF 624, you see this peak right there? That's indicative of a transmembrane domain. So if Ry Young was in here, he'd shake his head and he goes, yeah, the constipation hypothesis. This protein is screwing up the mitochondria, which is eventually, he would say, leading to pollen abortion. Okay. And then we have a series of other ones with the amount of time. I'm not going to go through them. You have an ATP6 with a novel N terminus. It isn't anything like the ATG at the start of the ATP6. And this one, we have a novel C terminus for the protein. So we came away with a lot of different candidates. Again, my opinion, the ORF 624 is the most logical one. So here's a summary of what we found. That's like about 6 years worth of work. Again, if with PacBio, it would be a month and a half. But ORF 624, as I said, it's an ATP1 fused with something else. And then we have a series of other ones. And those people in here, the reason why I don't think it's the novel N terminus, this gene that I have my finger on, my pointer on, actually exists in the A3 cytoplasm as well. Does anybody know a common fertility restore that restores both A3 and A1? Doesn't happen, right? So what I'm saying is if this is the real gene, but it exists in A3, the point we're coming away with is, okay, but I can't restore A3 with the same restore alliances that I use in A1. So most in the field feel, that's an interesting gene, but we don't think it's the CMS because it's specific for causing A1 cytoplasmic male sterility. It should only be an A1, okay? So we have our strongest candidate. So here we go. At this point in time, I do this, Shalis, you need to go here if you want to hike. This is Jackson Hole, Wyoming. But we had pretty much of a story at that point. I know I could have published it. So do I continue it and not dabble and spend more time hiking in mountains? You know, it doesn't mean I'm quitting, but it could be instead of holding a pipetman, I could be holding onto a backpack in Jackson Hole, Wyoming. Actually, there's a story about this where we saw a bear, but we won't go into that. That scared the hell out of me. So I didn't give up because we know fertility restoration genes are critically important in hybrid seed production. So what do we know of them? And I'll show you where we went next with this. First of all, they're all nuclear encoded. They belong to a very large gene family. And if you look at the sequence of these PPR proteins, almost all of them are shuttled into the mitochondria or the chloroplast. They don't exist in the cytoplasm. They get targeted. And with a lot of good work out of a lab, his name is Ian Small in Western Australia, they have found that these PPRs are actually RNA binding proteins. And you go, who cares? Well, what they think they're doing is they're going into the organelles and they're correcting the mutations. But the mitochondria doesn't have a system in itself to modify, to correct for defects like the nuclear genome would. So the nuclear genome is encoding proteins that go into the mitochondria to correct these defects. And it stretches everything from RNA processing, what I mean is cutting transcripts apart, but also something we'll cover in just a minute I call it gene editing. People call it RNA editing. But if you look at the sequence of the genome, and then you look at the sequence of cDNAs, they don't match. And I can't imagine the first graduate student that did this with the professor going, you need to go back and reread your sequence because that shouldn't be a T, it should be something else. But what's happening is these PPR genes actually edit the transcripts. They change C's to U's. Okay, and it's very prevalent, and it's one way that these PPR genes correct mitochondrial defects. And the very last thing I want to point out, it's a huge gene family. It's like the LRR genes that are fighting, that you talk about the race between pathogens and LRR gene evolution. And if you talk to some of the people in the area, they feel the PPR genes are in the same level of race with the mitochondrial genome. Alone in Arabidopsis, there's 400 of these gene members, okay? And almost all of them, if they're not a pseudogene, are heading into the mitochondria or heading into the chloroplast, okay? So that gives you what we've learned about these genes. So very first thing I thought, well, easy. we've sequenced the RNA. Let's look for editing. I'm guessing we won't find many. Well, I think we found like 400 sites within or 500 sites within the genome where we saw editing. And I'll just go through it. And what our thought process was as well, if these fertility restoration genes are actually doing gene editing, all I need to do is look at gene or RNA editing, excuse me, in the female and compare that to the editing in an F1 hybrid. Again, the F1 hybrid has the restorer genes. If the editing was associated with fertility restoration, I would expect to see a big difference in the amount of editing in the A line versus the F1s, okay? So regardless if we see editing where amino acids are changed, if it's the exact same edit in a restored line, then it's not very interesting. Well, it might be interesting to a basic biologist, but not for somebody that's trying to figure out what the RF genes are doing. So these here are going to be all the chimeric open reading frames. Again, what was I hoping? That I would see that the gene editing would be near 0 here and near 100 over here with the F1s. And that's not what we found. But we did find a series of edits. And as you can see, some of these are rather critical. A proline changing to a leucine is a critical amino acid change. And you go down here actually to the novel N terminus, there's actually the introduction of a stop codon. And you go, my God, it truncates the protein. It stops it. Well, look at the efficiencies. When we looked at all the transcripts within the mitochondria, do an RNA-seq, a very high percentage in all of them were edited. Okay. From a biological standpoint, I know we could publish this because if you look at the NAD1 subunit, this being the editing, this is the resulting amino acid. Without that edit, you wouldn't have a start methionine. And the NADH is a critical gene, protein. And so this gene editing taking place by the PPR genes actually established a start codon. In other cases, you see stop codons. But if you look across species, what they're doing is correcting mistakes. This protein is supposed to truncate after 80 amino acid. This one is supposed to stop after 438. If you go away from the grasses, if you look at Arabidopsis or something that's not related to sorghum, that's where they do terminate. So if you want to look at the editing, it's simple to look at. It corrects mitochondrial defects and it's conserved. And meanly, they're correcting lethal problems within mitochondria. So you can't look at it through all the looking we did not find an association of the editing with presence of fertility restoration genes. So strike one, can't finish the project on that note. But what a lot of people say is it actually relates to mitochondrial processing. And what I'm trying to say here is chopping up these transcripts. Okay, so you've got this toxic open reading frame. These PPR genes, the RF genes, will come in and slice that in two. Basically kills the chance of having a functional protein made. So what I did to try to do the most pertinent study, I think I finished these a couple months ago, 3 months, well, actually before we went to the field. And these are actually northern blots of looking at the transcripts, both from a B line where these genes shouldn't be present, the A line and the F1s. And we're just I'll put my cursor here because this is the easiest one to follow. And again, I talked about trying to get at this specific cell type. All of these RNAs are isolated from panicles right before how the anthers came out, okay? And so I was trying to get that stage of development that might show an effect of these RF genes in restoring fertility. And of course, you got to include the B line because if you see a signal there, you're in trouble because these genes do not exist in the B lines, in the normal cytoplasm. But the take-home message here, again, is there doesn't appear, if you follow this band from the female to the F1, the levels of the band vary. And that was just because it was very difficult for me to isolate mitochondria from panicles that they were at the exact same stage of development. But the unfortunate take-home from this is, in my opinion, because I thought I'd be done, is a lot of the literature will say, well, okay, if I looked at these three, that band would be completely gone, that would be gone, that'd be gone, that'd be gone, and that one be gone. So you go, there it is. The RF genes are actually chewing up transcripts. Therefore, this toxic open reading frame doesn't get made. But in A1 cytoplasm, in this endless project, that wasn't the case, okay? We did not come away with a feeling that all we had to do is look and say, okay, there's a northern blot looking at transcripts. We're going to see a change in the banding pattern in the presence of the fertility restoration genes. And we didn't. So again, a summary of that. RNA editing is ubiquitous in mitochondria. It doesn't matter whether it's in the presence of a fertility restoration gene or not. That's not how the RF genes are working. And we did not see a change to the second point in the transcript processing either. So it's not something as simple as things getting cut in half. The ultimate question that we start asking is, did we just miss a critical stage of development or a critical cell type? I mean, they have now evolved RNA-seq to work with single cell types, right? And we could do that, but in talking to the colleagues, trust me, I'm letting you out early. I'm not going to go on for another 100 slides, but just hang with it. So the Endless Project, and I like this slide because that was where we went to Italy, and I put it in here because you actually see plants in the background. So I thought it was relevant for crop science. Never mind. No, that's Italy. And I'm like, if I just quit this project, maybe we could go back to Italy so I wouldn't have the guilt trip of worrying about this stupid project. But anyway, so people that work in Arabidopsis would say, why didn't you ages ago? And they're right. Why didn't you just transform an A line with your candidate RF gene? Then you would have isogenic lines into which you can compare, really figure out what this RF gene is doing. And reason why I point that out is the way I'm looking at this, people who work in sorghum realize just because it has the RF genes in your restore doesn't mean the rest of the genome is identical between RTX 436 and RTX 430. That's not necessarily the case at all. Most likely it isn't. So when I'm looking at the A line versus the F line, F1s, I'm looking at a completely different nuclear genome. And therefore, how the heck do I really ascertain what the RF genes are doing with all that changes in the background? The early literature out of rice and that would say, oh yeah, just run these northerns and there it is. The low-hanging fruit experiments, okay? That's not how it's turning out in sorghum. So transgenic time, something that a Arabidopsis scientist would have done 15 years ago, but I kept waiting for the center here or somebody to develop a transformation system that didn't restrict me to RTX 430. But we're going to create isogenic lines. I keep trying to say near isogenic. They will be isogenic. And so what are we doing? There's a group of collaborators that have worked for years and years with the ballistic gun in Australia. The gentleman's name is Ian Small. He's a very good scientist, and the Australians have been working on sorghum transformation for years and years and years. And he has it now where I said, can you transform an A line, the embryos from an A line? He goes, sure, no problem. What do you want to do? So I'm going to work with Ian. I would love to work with Marco and Mayra here, but in talking to them, they say, well, we can transform RTX 430. Well, what good does it for me? I mean, they're very hardworking. They're very good at what they do. But for me to put a restorer gene into a restorer and then say, now I have to back cross out that RF2 into a different genetic background, I'd probably get it done in about 40 years. So the logic we're using is, let's transform the embryos from an A1 line, meaning we'll cross it to its B line. It's still sterile. Ian will take those green embryos, the seed from green embryos from green seed, and use that for transformation. And then the final thing is I've actually made the construct. This construct has been sent off to Australia. We're putting it behind a ubiquitin promoter. And those of you who know ubiquitins, the term ubiquitous, it's expressed everywhere. So what I'm hoping is that I can go in and work with seedlings. I can work with leaf tissue. I can work with roots. I'm not restricted to say I should be working with the panicle where this gene is most likely really expressed. If RF2 is being expressed behind a ubiquitin promoter, I should be able to look at any type of tissue. And actually, that's what they've done in wheat in Australia. Okay, that's where they finally determined what their RF genes were doing in wheat. They figured it out once they made a transgenic wheat by overexpressing their RF gene. Okay, so where does this project stand? Okay, we're getting to the end of this because ironically, the project I really wanted to present was the one on black sorghum, but the graduate student is still writing the paper, and I didn't feel that was appropriate until she finishes writing the second and last paper she's going to do. But that being said, I thought, all right, so I can do this cloning. And what we did is we got a vector. We're overexpressing the full-length cDNA behind the ubiquitin promoter using this plasmid, okay? But the other thing we're also doing, working with Juren Zhang's group, I'm making doggone sure with a C-terminal fusion to RF2 that we have an antibody to, that this thing is getting into the mitochondria. Because what if we annotated RF2 wrong? The actual reference annotation of this is completely wrong. They stuck in introns. These PPR genes don't have introns. So we went back in and re-annotated it. We're just hoping we got the annotation right. So my cDNA starts hopefully at the start of the transit peptide and ends at the stop codon, okay? So all good thus far, but Juren's group is going to actually just make sure using MitoTracker to see, does this recombinant protein get into the mitochondria? Because that's step one. If it doesn't, I'll have to tell the Australians to hold on, because if it doesn't get in the mitochondria because we did something wrong, there's no reason to go through all that effort. But you can easily transform tobacco protoplasts to do this study. The construct has already been sent off to Queensland. And again, the hope is that David and Emma have been growing up A and B lines over there. So when Ian Godwin wants the construct, he can just get embryos from them and transform. So hopefully about this time next year, they've got their transformants. What are we going to do with them? First of all, if this works, the A line better be fertile, right? Because now I've introduced an RF gene into a sterile background. And that's step one. Hopefully it'll say, yep, our tea plants made a lot of pollen. Therefore, we know we've correctly identified RF2, and we now have an isogenic system that will allow us to look at things. On my end, if I can get the seed back here, we're going to go back and look in our RNA-seq again. Because in wheat, when they did this, they said they really never saw what the RF gene was doing until they overexpressed the gene in wheat, their fertility restoration gene. And then they saw the processing that they just had a hard time seeing just using A lines versus hybrids. But something that relates to what I did on my postdoctoral appointment, there's another level of control. And I haven't done it this far because I want to use the transgenics. They're much more powerful. But what they're saying now, most people feel that the RF genes, what they're actually doing is they're binding to the transcript of these genes. And what they do is they prevent them from being translated into proteins. It's a classic translational control. And most of the work we did on chloroplasts shows that the organelle is basically controlled at the post-transcriptional level. You got the transcript there, it's just not making a protein. And we tried to make antibodies and I wasn't successful with those despite a great amount of effort to see whether I could see the proteins. The antibodies just didn't seem to work. So what did we learn? I'm summarizing, leaving a lot of time for bad questions. I don't want any good questions, I just want bad questions. Have you ever noticed that people say, that's a really good question? What the hell does that mean? I agree with you. I wanted you to ask this. I mean, never did understand. Good question. Anyway, massive genome reorganization in the A cytoplasm, otherwise known as in nuclear, people would call it structural variants. What's the big deal? That's where the cytoplasmic male sterility comes from, because you got these centaur type proteins, genes that are toxic, believed to be toxic to the mitochondria. When we looked at fertility restoration by comparing F1s to A lines, what did we see? Well, we didn't see an alteration in RNA editing or processing. However, onto this last line, I think we will see that with the transgenics, the isogenic lines. Not a guaranteed, but from the success of the people in Australia that did the same thing in wheat, hopefully we'll finally figure this out, okay? Now, you might say, God, that wasn't very much work. And what he presented didn't seem like a lot, but this is what happens. Two things: when your postdoc quits, who is going to do the project, which is irrelevant at this point, and when you dabble in a project, but also when you do a project when the technology isn't ready, like doing genomic selection or phenomics in an era 25, 30 years ago. It's better just to wait until those things become more routine. And I think if I had to redo it, I'd say, let's wait until somebody develops better long read sequencing besides phosmids and things like that. There was actually a very active running debate with the sequencing of the human genome. And the only reason why I know this is a group of us visited the Human Genome Sequencing Center at UT Southwestern, the medical complex. And they were using old technology. They were very important. That group was very important for an accurate human genome, but they had 25 or 30 ABI sequencers running 24 hours out of the day. And at nighttime, they would actually employ housewives. That's what they call them. But the kids would be asleep, the husband's home, the wives would come in at night and load samples on these machines. And then you had a group of bioinformaticians that would analyze it. That same amount of work now could be probably finished with a PacBio sequencer with 1 or 2 people in a far shorter period of time. But they wanted the sequence. And so it was revolutionary that they did that. But again, I guess in our case, I thought this was going to be easy. It was going to quickly identify the chimeric open reading frame, going to easily identify what the RF gene does. And that wasn't the case. But it probably would have been a lot less painful if we had just waited until the sequencing capabilities were present. So it continues at least for 1 more year. Then I get to go play with the grand dogs. The one here is actually completely grown up and chews up shoes really well. But they exist in Iowa, so I would have to take a nice vacation. But it continues for now because I want to finish this, partly because there were a lot of people who had influence or had a small part in this. And I feel like let's get a good paper out of it. We shuttle it aside. It won't revolutionize anything. It's not going to increase plant breeding, but by God, we'll know what's going on in the A1 cytoplasm. Okay, and for those that don't work in sorghum, A1 is the cytoplasma used by 99% of breeders. So it does have importance to a breeder, but me finding out what the RF genes is not going to revolutionize what you guys do on a daily basis. These are the people that worked on this project or the groups. I won't list names, I might mention them. It was originally funded by the Australian Research Council because David Jordan convinced them that if we knew where all the fertility restoration genes were, it would make his breeding a lot easier. So I don't know if it has, but they funded it. And so we did the work. And so over here, Queensland, that would be Emma Mace and David Jordan. But there was a little company at the time. Everybody had gone away from making back and mate pair libraries, but Lucigen was still dabbling in that. So I contracted with them to actually construct I supplied the inserts and they made the phosmid library. And then of course, Ian Small is the world's expert on fertility restoration. And his major emphasis is wheat, but I just look at what he does in wheat and it helps out a great deal trying to understand our system. And of course, AgriLife, there is no USDA farm here in this area. So I say thank you to Bill, to Trish, and anybody that allows us to do this research in this building and out at the South Farms. And that's it. So again, it was short. This was where I'd like to be right now. It's actually in a mountain. It's called Glacier Lake up in Montana. There's a nice hike up, but if you notice over in the distance, we got out of there pretty quick because that's not fog. That's a storm rolling in. So fortunately, we got back down. But anyway, folks, thank you. I know it was brief. Didn't have a lot of take-home messages. We didn't figure out what the RF gene does. But when Scott asked for one of our postdocs to speak, and I went, we don't have any postdocs and we don't have any graduate students left anymore. I thought, okay, I'll get the talk. So thank you for paying attention. And I was done.
I do have a question for you, Dr. Klein. It was interesting to see all that sequence of events. But we know also, to make it a little more challenging, we know that sterility sometimes breaks under some environmental effects. Knowing that, what would be the most plausible explanation then?
Right.
Sterility. Is it RNA based on the reaction to
It's
environment?
It's very much based, the way we talk about this, it's very much based on some change in gene expression. Now, does that mean that there is a decrease in the synthesis of these chimeric open reading frames? But you're right, because one of the things, if we could repeat, if I had a way of saying, I know when this A line is going to break sterility and produce pollen, by God, I just targeted it, and that's what I would have worked with. But my best efforts to get it to break consistently failed pretty well. I had it happen once on a tiller in a grow chamber, and I was so excited because it was like ATX 623 with just massive amount of pollen. I was like, oh my God, all I have to do from here on is to cut off the panicle, have a tiller come out and put it under high temperatures at night and high humidity. And I couldn't repeat it. But you're right, it breaks. It breaks. And whether that's a break in what's being expressed in the mitochondrial genome, I don't know. But I was like, this is a great tool if I could get it consistently to break sterility. We have one with the Arizona line, AZ 9504, whatever, that breaks the female breaks sterility when you look at it. And again, it was just a matter of, can I duplicate the conditions and where it breaks and consistently? So you're right, there's something changing, but we're very much interested in that. And that's another thing David Jordan was interested in, because he says, I'm getting tired of my females breaking late in development. If I knew what was causing, how I could track that in earlier generations, if we knew, I would be able to get rid of them instead of waiting for that off time 5 generations in, like I've heard with Daryl Rosenall, that beautiful A line and it breaks sterility. You throw it away. But if you knew that earlier, you could just discard it earlier. So it'd be nice to know.
I want to ask follow up on what you just said. So does sterility not break in earlier generations? Does it usually break at the end of the breeding process?
No, I thought it broke wherever, right?
I mean, the reason I was asking is because it could be related to stress. You know, inbreds are usually they're genomically stressed. And so if hybrids were more stable for sterility, then that may be somewhat of a key. But
Yeah. I've seen it, little I've seen it, it can break, but it's normally, if I'm correct, but again, you postulate it's high humidity, high nighttime temperatures, all this other sort of stuff. But it's really, for me, it was always inconsistent. So, you know, I did not see, most of the things I worked with were advanced lines. So I'll have to defer to Bill.
Another question about that, even if we do find something, there's probably 1,000 others.
Yeah.
All right, so you found one, so one of them, but that doesn't, I'm not sure. It's just like when you started this and we tried to use it for markers back in the 90s. Didn't work at all.
Didn't work.
Because there were so many other factors.
Right.
Now you know.
Yeah.
Okay, that's what's happening. So I still think that we stuck with the same
Right.
problems. You found one.
Yeah.
Out of 400.
Right.
Now you gotta find the rest.
Yeah, I won't argue with you. It's where we were getting philosophical, and it's something that is, all of us have pretty much changed our opinion. Back then, we all felt that if we could just map QTL and we can find all the partial fertility restorers and all the restorer genes, all we have to do is develop markers and we can get rid of those while making an A line. And then all of a sudden something else pops up under another condition and you've spent all that money on marker assisted selection. Just comes to the fact, this project has evolved into just to me, a scientific curiosity. And just, it's still interesting. It'll be more interesting with the isogenic lines. But the delusion that we're going to ever make this, in my opinion, where a breeder will say, thank you for doing this, it's really helping my program. I don't believe it. Sir.
So it looks to me like you're getting close to where you could manufacture your own, you can edit
Yeah,
and create your own result. Is that not?
that's absolutely what, see, if I believe in the statement, that's a good question, I'd say that, but sorry, it's a pet peeve of mine because that implies that they don't say it when you ask a question. It's not a good question. Excuse my sarcasm. Ian Small, University of Western Australia, is doing that exact same thing. He's the one that's figured out that the recognition site of these PPR genes, the exact sequence of RNA that they're binding to and editing. One of his major pushes is, by God, I'm going to go into the mitochondria and I'm going to start editing genes by making, as he calls them, custom-made, tailored, whatever, PPR genes. So that group very much is interested in using these PPR genes for doing editing of the transcripts. Now, again, that doesn't mean that's heritable either, right? You're editing the transcript, but it sure would be a nice tool for a molecular biologist. But
I just wondered, when you talk about the membrane, and the membrane,
constipation, yeah.
called it constipation. A lot of our herbicide technology is based on kind of almost conflict.
In the membrane, yeah.
And they developed a lot of modeling for proteins that will or will not.
Right.
How you get caught, but then
Yeah.
I think they call it one.
Yeah. There are a lot of software packages that will predict that. And there's some that are very well accepted and other ones that aren't as well accepted. And you get different results with the different, and it's just a matter of probably the most important thing. You'd have to go back to being a biochemist, a cell biologist to really determine whether they're stuck in the membrane. But yeah, I didn't know that about herbicides.
I have a question on the assembly. So you did show that the mitochondrial genome had some subgenomes. So I'm just wondering how you did the local assembly to make sure that you had everything correctly. for
Yeah.
those genomes.
So probably, let's put it this way, a lot of sequencing of the mate pair libraries, we actually had, I didn't go through it, we had mate pair libraries that are 2,000, 10,000, and 40,000.
Can you repeat the question?
I'm sorry. If online, if you haven't been hearing this, I apologize. I'm not used to giving Zoom seminars. The question is I think suffice to say is how do we know we got the correct assembly based on using the mate pair libraries? And how do we know we don't have it wrong? So the approach we took was different size mate pair libraries. So we would be covering different regions with different libraries. But you're right. When he had a discrepancy, he would say, I'm not sure about this region. And what I would have to do is do PCR. So all the junction points I confirmed with PCR. And it was sometimes rather difficult to design the proper primers, but he said this fragment is with this fragment in one subgenome and in another subgenome, they're this fragment with another fragment. I went in and did PCR across all those regions. But where it really made the big difference is the minion. That was like, we were still pretty unsure, but with a really nice long read sequence. I don't remember what the average insert size I gave them was 100,000 or some. It wasn't what you need for doing PacBio, but it was substantial enough that when these genomes some of them are only 80 KB, right? So it's not like they're 5 megabases long. The total genome, if you total it all up, is like 450,000 base pairs. But you're right, it was challenging and a lot of running PCR. And again, ultimately, we needed a much longer read to be sure that we were seeing all the correct assemblies.
So you're one of the first people I've heard give a talk on mitochondrial genomes. So really cool to hear.
Oh, thanks. I thought it was boring.
So you showed maize, and you obviously had to do a little background in maize, and the maize T cytoplasm
Yep.
caused all the issues. Does that same gene exist on the maize T cytoplasm in sorghum? Is there, the A1 is sort of a concern because of the T cytoplasm in maize.
Right.
Is there some lessons you've learned by looking at the genes that are on the cytoplasms in sorghum that maybe there's a fragility? That makes sense?
I'm lost.
Okay, we're in the heat building.
That part I got. I'll go back to this. So ask it again. I'm sorry, Seth.
So maize T cytoplasm cause problems.
Good question, Seth.
I don't.
I want bad questions.
Maize T cytoplasm cause problems with was it southern corn leaf blight?
Southern corn leaf blight. Yeah.
And there's different genes. You have maize, T and S cytoplasm on there.
Correct.
Now that you've looked at all these sorghum cytoplasms, are there complementary genes or any reason that we'd have concerns about just having A1?
Got it. What Seth, I think, is referring to, which was been a favorite project of Dick Frederickson years ago, when they found that the southern clearleaf blight wiped out a large portion of the corn crop, they realized it was due to the T cytoplasm. And a gentleman named Art Hooker was the one that actually somewhat figured it out. And he said, pull out the old maize with the normal cytoplasm. And with 1 year, they were back up and running again. But it was pretty devastating. What they found, it was called, has an open reading frame, TURF13. And the southern cornleaf blight actually bound to this protein in the mitochondria. It causes a pore within the mitochondria, and it just basically screws up electron transport. It's like two gears disconnected, okay? Because it was a nice little opening in the membrane. Didn't destroy the membrane, but it caused a pore. So once they found this, I know for a fact Dick Frederickson is like, by God, we got to find something similar to this in sorghum, because we could get a massive amount, not only from a pathology standpoint, you'd be curious, but you are going to have a really interesting project and some serious funding to work on it. And my understanding is they looked long and hard trying to find some disease that would affect the A1 or A3 cytoplasm, and they never found anything. And for what it's worth, that this open reading frame that causes cytoplasmic male sterility, and also is the thing that the Helminthosporium or Bipolaris matus toxin, a fungus attacked its toxin. It was specific for this gene as well. So there's a common feature amongst all the toxic open reading frames in the mitochondria. But if you try to say this one should look like what I found in maize, they're completely different. They just they're getting to the same place in the end, but they're completely different proteins. Besides, they're all fusions like a centaur, and they've got a membrane spanning domain. Beyond that, that's it. Right.
Other questions? You got anything in the chat there, Bob?
Oh, I mean, we can hear you. That's good.
My smart aleck comments, I'm not.
Yeah, I apologize.
So I got a question, Bob.
Uh-oh.
You're not gonna get your transgenic plants back for a year. How are you gonna retire?
I might not retire then.
You might not.
Can you say emeritus? So,
Yeah, you're not allowed to repair. I got a project for you in maize now.
do you really?
Oh, yeah,
Okay. You got big bucks to pay me?
maybe.
Ooh. But folks, thanks for listening. If I speak again, I would love to speak on the black sorghum once that paper's out, because I think that story is, to me, very interesting, has parallels with maize. And but this is something I've been dabbling in, and it's not the most interesting project. It's one of the most disappointing projects, because it was supposed to be easy, and it wasn't. But when we get it done, so be it. Publish it and move on. So anyway, thanks for listening.
Thank you.